special clip from Joshua Tippett.
My name is Joshua Tippett. Today, I will be teaching you about cell replication, transcription, and translation. This recording is for the BY210 class, Genetics, taught by Dr. Shahid Mukhtar. Ladies and gentlemen, today you will be seeing a romantically amazing rendition of what it really means to be inside of a cell. Actually, just kidding, it's pop. But it's good pop. Double-strand DNA, anti-pop parallel, 5' to the 3, helicase runs through me, complementary, gyrates, relays when stressful. Unzip your genes, no, not what you think, unwind the helix with this helicase, separate, cutting those base pairs away. 'Cause you know that, baby, I replicate them genes until they're synthesized in plenty. Make a lead and lagging. Leading strand is synthesized continually in 5-3. Lagging discontinually. Lagging means ligase. 'Cause Okazaki is fragment hey. DNA polymerase goes only 5 to 3, so leading outruns lagging.
Next up is transcription, or the process of writing down the genetic instructions to create a protein from DNA. The instructions are written as RNA, or ribonucleic acid. RNA is a polymer with nucleotides joined by phosphodiester bonds. The structure has a ribose sugar, phosphate, and a base. The RNA differs from DNA strands by using the base uracil instead of thymine to pair up with adenine. The role of RNA includes being the messenger between DNA sections and the outside world, or outside the nucleus, so that it can be translated into an actual protein. This type of RNA is called mRNA, and the process of going from DNA to mRNA is called transcription. So we have part of a DNA molecule, and it consists of part of a gene that will code for a protein. We create an RNA strand using the DNA in the nucleus as a template. To make the RNA strand, you need a promoter region, an RNA coding region, and a terminator. We go through 3 stages: initiation, elongation, and termination. In initiation, transcription will begin at the start site just beyond the promoter region, usually here. This base sequence is necessary to start. In bacteria, core RNA polymerase enzyme and a sigma factor will bind together to make a hollow enzyme, which will recognize the promoter region and bind to it. It forms an open complex that allows the RNA nucleotides to attach to the DNA template and start forming an unconnected RNA strand off of this parent strand. It goes 5' to 3', and initiation is over when the sigma factor falls away and elongation continues the process. RNA polymerase moves down the DNA strand and the DNA is rewound up. It is terminated after the terminator sequence has been transcribed. This can be row-dependent or independent, depending on the organism. For row-dependent, a row protein will attach to the RNA strand at the row recognition site, and a loop will form between this and the termination sequence that will cause everything to stop. The row protein will move down the RNA polymerase, and the RNA strand will dissociate from the DNA parent strand. In eukaryotes, the process is similar. Initiation occurs and modifies the chromatin structure. There are different types of RNA polymerase that recognize different promoter sequences and use different mechanisms of termination that will release the RNA polymerase from the DNA strand and the RNA to come off. What is left after this process is this: an RNA single strand with a 5' guanine cap and a 3' poly A tail. These extra nucleotides make it easier for the mRNA to leave the nucleus and help protect it from other enzymes that are present. But before leaving the nucleus, the RNA has to get rid of the extra information that has nothing to do with making a protein, and this process is called RNA splicing. We first use SNRPs to signal the start and end of the splicing areas, and we then use spliceosomes to actually do the editing and take out the introns that don't express anything. This leaves the exons present, which will express everything needed to make a protein.
How does an mRNA lead to protein synthesis?
So next we're going to look at the second step of protein synthesis, which is called translation. There are 3 steps of translation: initiation, elongation, and termination. The small subunit of the ribosome made of rRNA finds the mRNA for a specific sequence called the Shine-Delgarno sequence and attaches at this point. After attached, the ribosome then scans the mRNA for the initiation codon. At this codon, a tRNA charged with the amino acid FMET and its anticodon is attached. This attachment then calls for the large subunit of the ribosome to attach, thus making the initiation complex complete. Another amino acid charged tRNA with the appropriate anticodon attaches to the A spot, codon of the ribosome. Then, the amino acid that is attached to the tRNA on the P spot forms a polypeptide bond with the amino acid on the A spot. The tRNA that no longer has the amino acid then moves to the E spot, and then the tRNA that has the polypeptide bond moves to the P spot. Then, the tRNA that's in the E spot exits as another complementary tRNA enters on the A spot. After the chain is done elongating and a nonsense code is reached, a release factor is attached to the codon and the entire complex is separated.
Is there a difference between eukaryote and prokaryote translation?
In eukaryotes, only methionine is the initiation tRNA. Also, in eukaryotes, the small subunit of the ribosome scans for the Kozak sequence.
DNA replication occurs, and how protein synthesis occurs. Thank you.
No problem. I'm glad that you learned something.
Some legends are told, some turn to dust or to gold, but you will